Could alien life have sparked on worlds beyond our own? What might lie just beyond the horizon? For over a year, world-class scientists have simulated alien life forms. Life that is strange, yet familiar. Join us in this adventure to explore what life could be. The Berggruen Institute presents Proxima Kósmos, Journey to Alien Worlds.
Hi, everyone. Welcome to everybody, the researchers who are on the call and all of the guests who are attending this virtual symposium. I'm so excited to share this project because it's the first time that all of the researchers are together and presenting it to you guys. So I'm going to just do a quick little introduction and then we can move on to the planets, okay? So Proxima Kósmos, it's Greek. So you might know that Proxima Centauri, which is the nearest star and planet system to our own, it means near, but it also means next. Cosmos means world. So Proxima Kósmos carries a lot of hope in it that we will find life-filled worlds soon. And today it feels more and more, it feels like we're just on the edge of some kind of confirmed detection. And in our own solar system, we're discovering new kinds of worlds, even locally. So it's a really, really exciting time in exoplanetary astronomy, and really raises a lot of philosophical questions about what is the category of life? What will it mean to find a biosignature, a sign of life? Are we looking in the right way? And how can we reframe research questions that are maybe conceptually different than we have in the past? So I'm so excited to have you all. The mantra of Proxima Kósmos is speculative yet plausible. So the worlds that you're going to see shortly are, yes, I think they're gorgeous, thanks to our amazing design team. But the life that the principal investigators have simulated and evolved on their planet, sometimes over billions of years, they really could exist based on what we know of the current laws of physics and biology and chemistry. So I think that, it's a gorgeous project of imagination, but it's also really robust scientifically, and we're going to share as much of that as we can today. So xenia is another Greek word. And the translation now is an alien. Xenia in ancient Greece was a custom of you would let in a traveler from a faraway land. This traveler could perhaps be Athena in disguise. And so you always had to, it was like a sacred insurance policy. So you would let in the stranger in exchange for applying them with wine and giving them a nice meal. They would then exchange with you news of the outside world. And I love that Xenia references an alien, but kind of in a different way than we're used to. Aliens are strangers waiting at the threshold and I just think that that's like a beautiful way, not only to think about exoplanetary astronomy, but in this current moment of deep uncertainty with politics, I think it also applies. Okay, so just a quick housekeeping. Thank you so much for everyone attending. Thank you so much to the researchers who have been involved for years at this point in developing these amazing planets. And yeah, we're going to see 7 of these worlds. Each presenter is going to give a short talk about their planets, going through their research question, their hypothesis, the methods, and then what they discovered. This is being recorded. We will not be doing a Q&A session. However, do put your questions like in the chat because we're going to save all of this, and we're really interested in what you think. So we welcome commentary. Okay, let's explore alien worlds. Okay, so Sarah and Yarrow, do you want to say hello and take it away?
Hello, everybody. I'm Professor Sarah Seeger from the Massachusetts Institute of Technology.
Hello, I'm Jaroslav Jakubowski, or Yaro. I'm also working at MIT.
We're here to tell you about the planet Phaenoterra, which is phosphine Earth. And this planet has been inspired by Venus, although it's not Venus. And it has also been inspired by the very controversial detection of the phosphine gas in the Venus atmosphere. Phosphine is highly reactive and shouldn't be there. If it's still controversial, but it has motivated us to study not just Venus, but to imagine exoplanets like Phaenoterra that might have phosphine in them. Now, it turns out that Phaenoterra has no water, and all life on Earth needs liquid water to survive. But this out there idea is that perhaps other solvents, other liquids could support life. And in this case, we're talking about sulfuric acid, liquid in the clouds and perhaps on the surfaces of Phaenoterra. What's so great about Venus is that we can go there. And what you had just seen was a balloon, because part of our research involves studying missions, building instrumentation to go to Venus to search for signs of life by way of very complex molecules. So not phosphine itself in this sense, but to go to Venus and use a mass spectrometer and to collect the cloud particles and to look for molecules so complex that they're highly suggestive of life being present. And this shows you, this rendering is part of our balloon mission schematic. But you can see here, it's an alien landscape. It's a very toxic, scary world, actually. And Phaenoterra is incredibly mysterious and amazing. And what's so interesting about our work, motivated by Venus, motivated by Phaenoterra, motivated by phosphine, is we have been doing laboratory studies. And we have been demonstrating that although our Earth life can't survive, many, many biomolecules can survive in concentrated sulfuric acid. And these include our amino acids, our 20 biogenic amino acids, with one exception, are stable in concentrated sulfuric acid. Half are chemically modified, but stable. We have found our nucleic acid bases, that's the latter part of the DNA, ACGT, stable, protonated but stable. We have been working towards a DNA-like molecule, not our DNA, which is unstable in concentrated sulfuric acid, but like an analog swapping out parts, not to prove there's life on Venus, but to show that it's worth going to Venus with the balloon mission. And we have recently shown PNA, peptide nucleic acid, a single strand at relatively moderate temperatures is stable. So much more to tell you about our laboratory work, but I am going to turn it over to Yarrow.
Yeah, thank you. So I've been trained as a space technology engineer, so I typically build instrumentation for space missions for planetary investigation. But what's very different about Venus and Phaenoterra and such, because you need to test your instrumentation and you need to verify it. And you end up doing a lot of chemistry and a lot of biology, and you grow cells and you do mix various things in use instrumentation that you are not trained to. And this has been very educatable and very interesting experience along to do the experimentation in laboratory in order to inform our instrument and define measurements we need to take in order to find life or signs of life in other worlds. Here, it might look to you like a Venus, very toxic clouds surrounding you, but in fact, it's not. It's me in the field trip in Indonesia. It's the largest sulfuric acid lake on Earth, on the top of active volcanoes that was just erupting a few months before that. And around it is SO2 gas surrounding me, just like would exist on Venus or Phaenoterra for that matter. The planet with active volcanoes that emit gases and has a basic organics that has been found and was spread around asteroids and comets in our solar system. So it's very interesting to go beyond the lab where you have a predicted a few narrow compounds and uniques and have a very knowledgeable outcomes because many papers have been done and people doing it all over again. But once you go to nature, what's different? It's very complicated sample because it contains so much information in even microgram of a liquid collected even in a very acidic lake, like as one present in Aegean, which has acidity close to the one found in battery acid. And in order to distinguish what we can find in the nature, we have to go and collect the sample and use the instrumentation and train our algorithm to do so.
We've talked about our mission design. You saw the balloon rendering, which we hope will go to Venus in the next decade. We've talked about our laboratory work, and we forgot to mention those gorgeous images were from a scanning electron microscope. And Yarrow just talked about his field work to collect samples. And Phaenoterra has motivated us to even push beyond all of that. And here you're seeing an artist's rendering. Now, on Venus, the surface is too hot for sulfuric acid or any liquid that we know of, but it's the clouds where, if there is life, that's where it would be. But we're taking the advantage of Phaenoterra to imagine that there are pools of acid. Now, what is still under debate is whether sulfuric acid will destroy rocks. And indeed, I wanted you to know that concentrated sulfuric acid, so say 98% weight by weight, the rest water, behaves very, very different chemically than sulfuric acid mixed with water. And so nonetheless, because the jury is still out on whether or not pools could accumulate without the sulfuric acid seeping away or destroying rock, we're now envisioning Phaenoterra to have these plateaus in the mountains where the clouds, much lower than the clouds of Venus, lower in altitude, are coming by, and these sulfuric acid clouds could continuously replenish these pools so that life, very primitive life, we're not thinking about fish or dolphins or octopus, but we're thinking of really, really simple life forms might exist on our conceived hypothetical Phaenoterra. Here's another real film from Yarrow's Aegean field trip. At this point, we would like to thank Claire Webb and thank the Berggruen Institute for inspiration, motivation, and for sending us on this incredible journey that they've partnered with us on.
Yeah, thank you so much for letting us imagine worlds that are so close and familiar, but at the same time are so alien. Thank you.
Thank you so much. The appreciation is on both sides, as you both know. I want to make a comment on just how fun this project is. There was, if you saw the figure kind of sitting within the clouds, that was Yarrow at an analog site in Indonesia. Analog sites are often a way that scientists like Sarah and Yarrow explore alien worlds and kind of conjure their materiality through like environments on Earth, and it's a really generative and productive way of exploring alien life on Earth. So thank you so much. Okay, so now we're gonna go to a planet, Newstera, in which the scientist Lee has imagined intelligence and consciousness in a non-carbon-based framework. So we're carbon-based and we're squishy and from organic material, but on Newstera, intelligence begins with light. Okay, Lee Cronin.
Thanks, Claire. It's great to be with you guys. So I'm an inorganic chemist. I'm a Regius Professor at the University of Glasgow, and actually my team is kind of dedicated to uncovering inorganic evolution and getting to biology as we know it and as we don't know it. And I thought in exploring the scope of life in the universe, I wanted to do something a bit different and kind of think about what other causal mechanisms could there be to create complexity and information processing and evolution. And I think the driving question is, could we get evolution to emerge in ways that don't just go beyond the chemical bond? And I did this to challenge myself because obviously I think that chemical bonds are vital for biology as we know them. And we just heard from Sarah and a colleague about how looking for interesting molecules is going to be probably the key to finding life. So I wanted to go a bit beyond that. So how could we go beyond to a kind of optical technology or an optical biology? So the idea here is that maybe this is kind of inspired by Solaris, if you like, but grounded in well-defined chemical and physical processes. So if we go to the next slide, I can explain what we want to try and do, because if we're going to make a bizarre life form, we need to have a method that's going to allow us to detect life, whatever kind of manifestation it takes. And remember, as a chemist on Earth in Glasgow, I'm trying to build molecular-based life. And in doing that, I developed a new theory with my colleagues in ASU with Sarah Walker and her team to explore complexity and count the complexity and find out what life is in a way, in a more general sense. And what I would say is that this method we've developed called assembly theory detects systems capable of generating complexity at scale. This is what biology seems to do on Earth. And we wanted to see if Mind Earth, Nostera's atmosphere, could also kind of get to complexity. And so we wanted to imagine a situation where assembly theory could be used to track the emergence of complexity across photonic structures. Yes, we can apply it to complex molecules, and we'll be doing that in the solar system beyond. But what about developing a new type of device to look for complexity in its most general sense, not just molecular, not just material, but optical? And so in this case, we wanted to kind of build a system that would capture that. Before we say that, I just want to give a little bit of background to what assembly theory does. Assembly theory basically says, hey, if complexity is kind of weird, right? The universe will not build complexity unless there's evolution or technology. At first glance, this statement seems outrageous, but actually what we see beyond Earth is just complicated and not complexity, because complexity requires recursive structure. And the way that chemistry gets this is through bonds, building up more complex molecules, amino acids, DNA, RNA, and so on. But what we wanted to try and do here is to say, well, could we simulate a similar type of architecture, but rather than using chemical bonds, using the force of light to join together lenses, so we can have this architecture that's self-lens and take light and put it into some kind of cavity to create an optical force analogous to the chemical bond that will allow us to make a life form. And in a way, this is kind of cool, because this allows me to challenge myself, because I was saying to people, well, of course, you need chemistry and bonds before you can have cognition, consciousness, and all that stuff we like to think about. But I thought, well, let's turn the tables. Is there another way that information processing could give rise to life, replication, and technology? And this is the kind of attempt to kind of ideate that out. So how do we detect it? So the assembly theory has this equation that we can use to find organic and inorganic life. And I think that this will be covered a little bit later, because there might be a way we can look for complex molecules in planetary atmospheres. But imagine you have your life meter and you were scanning. What would you be looking for? Well, the assembly equation has three components. The first of all is the assembly, big A. That really literally tells you how much selection is going on in the focus point. And this just literally measures at a distance the complexity of the molecule, number one, how many parts, and how much are of there. And what you can see in this meter is the orange A is only above the threshold if the molecules, or in this case, the photonic nanostructures, have a number of parts that are non-trivial. And there is a high copy of them, a large population of these photonic bacteria. And so this measures this complexity through light and structure, not cells. And this allows us to imagine a way that we could remotely detect life on a planet like Nostera. So what we want to try and understand is, like, in the system, how did life even emerge, right? We can imagine chemistry happening in a Darwinian sense, in a Darwin's warm pond, or some kind of origin life experiment. But maybe we can imagine where there would be some kind of interplanetary kind of excitations, gravitational movements, lensing, focusing light. So the light and the gravity could affect the atmosphere, where you've got these lenses which are light enough to float in the atmosphere and trap light and make optical tweezers, and a series of cavities that could self-organize and take the light for replication. Again, the light gives the bond gives the causal structure. The causal structure gives the ability to generate a complex object that we could call alive. So this is kind of where we'd imagine the new worlds, right? This is kind of in the Sarah, life is emergent in the atmosphere, because the flakes of lenses are able to kind of float around, but be trapped in optical cavities and process information at the same time. And you could almost imagine the elements on this planet could move light from one side to the other and use it to process information. And remember, Darwinian evolution generates complexity, and that complexity has to survive and propagate. And assembly theory allows you to count that complexity and now measure it. And I wanted to imagine where would be the next level to go to consciousness in photonic systems. Could we generate high assembly structures that evolve beyond traditional inorganic and organic chemistry? And can we imagine an optical meter for finding life at a distance? Because that's going to be one of the ways we find life on exoplanets. And that is my planet.
Thank you, Lee. Okay. What's also one of my favorite parts about this project is that researchers like Lee can explore really radical ideas in this imaginary space of and scientific space of speculative yet plausible, and really kind of challenge what's familiar to us and humans, as a way to kind of go beyond the Terra-centric thinking that sometimes stymies us. Okay, thanks, Lee.
Thank you, and thanks for your help, Claire. It's been amazing.
Yeah. Okay, so now we're going to go to Acrostera. This is extreme Earth. So this is by Dr. Penny Boston, who's an astrobiologist, and she has, from her deep research of being a speleologist, one of my favorite words that I now have the occasion to use a lot, thinking about collective intelligence within alien caves. Okay, thanks, Penny.
Thanks, Claire. It's delightful to be able to talk about this fun project. It's been on my mind for quite a long time, like everybody. And it really is the coming together of a lot of my early work on extremophile organisms. And then in mid-career, finding that I could pursue that in the caves of Earth, many of which are far more different than the average tourist cave that you might have been in. It's very clear that you can form cavities and fracture habitat within just about every rock type that there is, including ices and rock salt and lava and the more conventional types of rocks. And so early on in the 2000s, I tried to formulate a way to think about how you would make, some sort of habitational array that could be exported from what we know here on Earth. And those cave forming processes and fracture forming processes, how would they manifest on the radically different planets that we have in our solar system, and perhaps someday beyond. So that's the theoretical framework that I brought to this work to begin with. There are a number of themes that I have found fascinating for a long time, including the fact that on Earth we have some types of life that have evolved to have a very different way of living. So, we all learned in school that life is essentially composed of cells. And this is true for the vast bulk of organisms on our planet. But there are some organisms that dispense with cell walls and have a big membrane within which there are many nuclei with the genetic information molecules, and they're free in the cytoplasm. The cytoplasm can be highly organized, and parts of the cytoplasm become gel or become soluble, and so that creates structure and channels within some of these organisms. You may have heard of slime molds. These are one of the organisms that exhibit this. There are also gigantic algae in the ocean, and some of those have what's called a syncytium. That's this notion of having no cell partitions internally. And what that allows one to think about is those organisms, even at the larger scale than we experience them here, being able to move in a way analogous to an amoeba, but at a much larger scale. So that's really fascinated me for a very long time. My planet is really cold. And even though a lot of the work I've done is at nominal temperatures on Earth or very, very hot temperatures, I've also worked in ice caves. The cave that you see on the right is one of the very hot caves with these gigantic crystals. And you see the team is all suited up in little orange suits, and you might think that it's a cold system and they're trying to keep warm, but actually they're packed in ice with refrigerated breathing gear because this is a very, very hot system. One thing that's really important about studying caves on Earth as models for extraterrestrial investigations is that depending on where on Earth a cave is, the internal temperature may be very uniform, but you can pick caves on different places of on the Earth where they can be either very hot or very cold or in between. Many of them have exotic gas mixtures that is very different from the air that we breathe on the surface. And so all of these are like an entire extra set of solar systems just based on our own planet, where we can test some of the ideas about how life can push the envelope of possibilities here on our planet and give us inspiration for going elsewhere. So what we know about caves so far on other planets is relatively rudimentary. We know that on Mars, there are very large pits. There's some examples on the left-hand side here. These are formed analogously in Mars's history, and indeed the moon, and actively today on Io, one of the moons of Jupiter. And these form very hot channels within lava masses. These eventually drain out when the eruption ceases and is no longer feeding lava into the system. And it produces these lava tube structures and pits in volcanic places all over Earth. And even though they're open systems, the conditions within them are radically different from the surface in which they occur. And so they provide very extraordinary circumstances for organisms to adapt. If you can see the lower right-hand corner, I don't think you can see it tremendously well, but that shows a cooled lava tube with microbial colonies that are hydrophobic. And so the moisture condenses on them, and they look like glittering fairyland in a lot of these places. So you can even see where microbes are individually very tiny, but when they are there in the aggregate, they can be quite macroscopically visible.
Penny, you got 3 minutes per request.
Yeah, that's great.
Chiming in. Okay, great.
Great. So one of the lessons that we have learned that I tried to bring to this planet is the fact that in caves, microorganisms are not the boring spheres, rods, and spirals that we think of from our studies on the surface, but they have very elaborate three-dimensional structures. There are a few examples from our work that you can see here. but I wanted to fit the idea of the life forms that I wanted to create to at least a semi-plausible chemistry on an extremely cold planet. So Acroterra is exceedingly cold, in the range of temperatures that you would expect on Titan, Saturn's giant moon. And so I posited that we were looking at organosilane compounds. So silanes are an analog of organic carbon-containing chemistry. The problem with a lot of silanes is that they're in solid state or vapor. So that makes it harder to make biology out of. But when you mix them with carbon and produce this mixed set of compounds, then you get at least maybe a plausible set of molecule possibilities to build life. So that was to accommodate the very cold temperatures. Next, please. But what I really wanted to delve into is this ability to connect the surface and the subsurface of a very challenging environment. So you can see on the right-hand side and in the center, this is a fumarolic ice cave that occurs in the vicinity of Mount Erebus, an active volcano in Antarctica. And these caves contain all sorts of interesting microscopic life forms. And so that was another one of my models. And I wanted to connect organisms that might be able to live on the surface with the subsurface and find the way to have an organism type that could make its way through caves in a way that we don't actually see on Earth. Next, please. And I based this on plasmodial slime molds, which I mentioned earlier. You can see on the right-hand side on the top an image of an actual budding plasmodial slime mold. These have been studied for their ability to actually have a type of intelligence and make decisions. The earliest example of that was published by Nakagaki in the early 2000s, showing that slime mold can solve mazes and go from one food source to another. So taking an idea from one of my favorite science fiction writers, Werner Vinge, who developed the idea of incremental intelligence of pack organisms, I wanted to apply that to increasing incrementally sized plasmodials and have them flow through the caves, have them be able to disaggregate and pioneer other areas, and then come back together again and become gradually more intelligent and decision-making organisms as their planet evolved through time. Next, please. So there are many, many details I haven't gone into here. I think I've had a fantastic time playing with this, and I'm not done yet because I've got a number of questions that I wanted to look into. But I love my Acroterra planet, and I wish I could go to one sometime in my lifetime. But the second best thing is to imagine it and go there in your imagination. So thanks to Claire and her team and the Berggruen Institute for getting this kind of thing and allowing me to play for real. Thanks.
Thanks so much, Penny. I was reading about how they're using slime molds to redesign the Tokyo subway.
Yeah.
Yeah, because it's like getting, it's like the
decision.
most efficient way. Yeah. Yeah, it's so cool. And it just is a reminder of how capacious the term intelligence is and how it's just it's part of the discovery process of trying to detect alien life is reconsidering concepts like consciousness, intelligence, alongside life in an extraterrestrial context. Okay. All right. So next up, we have three co-orbitals. Aponorostera, early Earth, Necrostera, dead Earth, and Xenoterra, strange Earth. So there's that Xeno word again, the strange but familiar. These planets are all versions of what Earth could be, or what it was. And instead of instead of biosignature, signs of life, Necrostera is a portent of what we might be evolving to, and it's a future that we don't want to happen if we wanted to continue flourishing flourishing on Earth. Okay, so Sam Krigman, who's at yes, he's right there. Hi, Sam. What is so fascinating about his work is that it is flirting with not exactly Darwinian models of evolution. So what I mean by that is that in these virtual worlds that Sam has created and the creatures that have evolved within them are based on like a determined behavior ahead of time rather than randomly evolving. Okay, thanks, Sam. Go ahead.
Hi, thanks, Claire. Can you all hear me all right? Okay, great. So I am very fortunate to get not just one new kind of planet all to my own, but three. And actually, the way that I like to think about these is that they represent an infinitude of like possible worlds. They're all Earth analogs, as Claire mentioned. One of them is early in geological and thus evolutionary time. One is very late in its geological time, and one is somewhere in the middle or closer to our own. What would life look like on some other planet? So I have this driving question here of what are the universal laws, if there are any, of adaptation, life, cognition, maybe sentience? If we replayed, fast-forwarded, restarted this movie of evolution on other planets, my hypothesis, and I share this with many others, is that the universal laws of animals, which might be modularity, symmetry, certain kinds of symmetry, legs to reduce friction on terrestrial surfaces, etc. These kinds of things that emerge again and again, these are the important details of life. There are going to be local, non-universal, superfluous details that just so happen to occur on our planet or other planets due to frozen accidents, and things that, while interesting and important, are not necessarily evolutionarily important. So here are some different kinds of alien creatures that are just shown here, and some might look familiar and some might look more alien. For example, one has 4 legs, like a quadruped, 4 limbs. And we also, and all terrestrial vertebrates have 4 limbs. Now, is this something that we should expect to see on other planets? Why is it in movies that aliens always look like us? Is that correct? Will there be bipedal aliens with language and these other things on other planets? Or is that just kind of an accident of evolution? Could we be sitting here with 6 or 8 limbs if it was a different kind of fish with a different number of fins that crawled out of the seas 400 million years ago? So this is the kind of stuff that I'm very interested in. If I had to reframe this driving question, I would just say, what does an alien look like? I'd love to know. I mean, who wouldn't? Obviously, we all would love to know that. I would jump on an interstellar ship now if it was safe and I wouldn't waste my whole life. I would jump on a time machine right now and quit my job if I could and explore these questions, since those are not possible as far as I know. I unfortunately have to be a computer scientist and roboticist, so I'll tell you about my work here and how we are simulating alien life on these 3 planets here at home, and not just simulating, next slide please, but also transporting these life forms back to Earth. And I know the video is a little bit choppy, but you get the idea that these are some dynamic creatures moving here on our campus at Northwestern. Some, as I mentioned, look very alien to us, and some may look more familiar. Each one of these modular units has its own brain, metabolism, and muscle. And so unlike obligate multicellular organisms on our planet here, the parts of these aliens remain truly independent, like some modular marine invertebrates here on our planet. So there are things that these aliens that we've transported back to our planet remind us of ourselves and of familiar animals, but there's some differences here. And we can explore, as Claire said, as these are evolving. Does it have to be the case that Darwinian evolution is the only way? Or could other kinds of evolution, like Lamarckian evolution and things that are more directed, could those work? Or are they doomed to failure? Is Darwinism truly universal? This is something we can try to get at through robots and through simulations on computers. Now, those kind of might have been boring. They were exciting to me, but maybe boring to others that wanted to see more anatomical complexity. So on our planets, we're trying to evolve more kinds of intricate internal skeletons and soft tissues surrounding these, and seeing what kinds of shapes and behaviors emerge with minimal assumptions. So here, it's kind of like Lego. You can build up a free form alien from scratch without having to say ahead of time that it should look like a dog or a human or anything else. Whatever will do the task will evolve in the simulator. And of course, these are somewhat barren, desolate petri dishes that we've transported these creatures from our planet into on our computer, starting to get a little bit more complex with different kinds of terrains and objects, but we still have a ways to go in beefing up the simulations to really be called not just a simulation, but a virtual world and recapitulation of these planets that we're studying. What's really great is that we have full control over everything. We can peer into their DNA and we can adjust their genes. So this is the gene, the collection of features inside of their genotype or their genome that changes their volume. So if you look at each row, the same anatomy is just getting more, is getting thicker. And we can find genes for just about everything in there: height, weight, width, how many bones they have. So we really can do genetic engineering and try to maybe understand some of the challenging aspects of that without having to harm any living creature. And yes, we can move through this entire genome space, interpolating between different possible forms and really getting kind of a bird's eye view of the possible alien forms that could exist and be well adapted to each of these different planets. Okay, so this is just kind of a teaser. These planets are still under construction or under evolution, I should say. So the question is, what else will evolve inside of the other unique habitats in water and volcanoes, in caves on our 3 alien Earths? And I don't know the answer. I have hypotheses, but we'll just have to wait to find out. Thank you.
Thanks so much. Thanks so much, Sam. All of these, every time a researcher talks about their planet, I'm like, that's my favorite planet. No, that's my favorite planet. And so it's really wonderful. Something that I learned recently because of this project is that Earth's early oceans were not exactly blue before chlorophyll was introduced. The oceans were kind of like a rosy pink based on geological traces of just a few billion years ago. So I love to like reimagine Earth as being kind of this like Barbie pink color. Okay, so thanks so much. Okay. And last but certainly not least, I want to introduce Estelle. Estelle and Sarah Walker at ASU have constructed Magicos, which is Magic Earth. And what is kind of amazing to me about Magicos is how Sarah and Estelle are thinking about intelligence as at a planetary scale. So there's this biosignature that we're familiar with on Earth. You could say that the network of satellites and underground cables, and the International Space Station, these are all objects that signify that Earth is moving from a biosphere to a technosphere. And Magicos takes the next step of that. And I'll leave it to you, Estelle.
Thank you, Claire. Yes, hello everybody. So Magicos is a world that Professor Sarah Walker and I imagined and worked on. And what we really tried to do is to illustrate the implications of complexity generated at a planetary scale. And we're particularly interested in the sorts of evolutionary transitions that would be associated with a world that is as complex and advanced as Magicos. And in this context, Magicos has transitioned from a biosphere, so a world where only biology is present, to a technosphere where an advanced civilization has emerged and technology has kind of fused with biology in a sustainable and symbiotic way. So in that way, you can say that life and planetary intelligence have become indistinguishable. And it is now on the cusp of a new evolutionary transition that we describe as planetary reproduction. And I'll try to explain what that means in the next few slides. So our driving question was, if we apply the principles of assembly theory, which Lee Cronin already explained a little, at the scale of an entire planet, what would be the implications for life and intelligence as planetary phenomena? And so assembly theory predicts and describes how evolving systems grow in complexity through deeper recursion and selection. So the way we quantify this complexity is by counting the minimum number of steps it takes to build an object. And the more complex an object is, and the more abundant this object is, then it means that the universe must have stored the instructions of how to build this object. It cannot just randomly fluctuate into existence. And what we mean by object in this context vary a lot in terms of scales and systems, and Magicos itself can be considered as an assembly theoretic object. And in the context of this planet, Magicos has become so complex that the planet itself can be considered alive. And in this construction process illustrated by assembly theory, the planet is ready to become a building block itself in the forward construction process of the next version. So Magicos and baby Magicos. And that's why we say that it's capable of reproduction through the birth of baby Magicos. This is illustrated on through the diagram on the left, where you can see that Magicos is able to probe deeper in its past lineage, so the recursion is much deeper in order to build Magicos and baby Magicos further in that causal cone. Now that's on the kind of conceptual speculation side, and Sarah and I were very interested in trying to understand what would the observational signatures of a planet as complex as Magicos be. And our observational window for these remote worlds are their atmosphere, and more precisely, the makeup, the gaseous makeup of the atmosphere. So which species are present above a certain abundance threshold. So we applied assembly theory to the chemistry of planetary atmospheres, modeling this time chemical bonds as the building blocks to build the entire atmosphere as the assembly object. And we simulated many different planetary types, but in this context, I'll introduce 5 of them. The first one is a lifeless planet, so a dead planet where life is not present. The second one is one that is akin to Archean Earth, where you have isolated ecosystems scattered on the planet, where you cannot really talk about life as a planetary scale object yet. The third one is modern Earth, so where global biosphere is present, but no technology. The first one would be what we are today, so a biosphere with a technosphere and technosignatures present in the atmosphere. And the final one is Magicos and baby Magicos, illustrating a much more advanced and mature technosphere. Okay, so this slide is a bit of a mouthful, but basically what we showed is that when we analyze Magicos's atmosphere through assembly theory, it is consistent with a very advanced technosphere. And the diagrams that you see at the top are what we call metro maps, and they allow us to visualize the assembly space of each atmosphere. And these assembly spaces correspond to the shortest path, so the minimum number of steps it takes to build all these atmospheres. And this allows us to quantify the complexity and the depth in time of these planets. And it's a bit more complicated than this, but basically more interconnected trajectories is consistent with a higher complexity and more selection evident in the chemical space of these atmospheres. And what the bar plot on the right shows is that compared to dead planets, Magicos is measurably distinct through its assembly index. And also, we're able to capture a continuum in complexity between different evolutionary stages, going from Archean Earth to modern Earth to Magicos. And the narrative that we're trying to introduce is that assembly theory, as applied to atmospheres, functions as a compressed signature of deep planetary selection. So in other words, the complexity generated at a planetary scale by a biosphere or a technosphere translates into measurable properties of the chemical space of these atmospheres. So we worked with artists to try to illustrate what this planet would look like. And what we came up with is that basically the surface of Magicos would be pulsing with the memory of its evolution. So matter is shifting, building, and the planet itself is alive and restructuring matter to build the next version of itself. So it leads to techno-ecological systems that are visible from orbit. And evolution and complexity has reached a threshold where a planet can remember and replicate itself. So as I said, the planet is now so complex that it has stored the information of how to build itself, and that's how baby Magicos can be born. Okay, so in conclusion, Magicos suggests a new paradigm where life can be understood and characterized as a planetary scale object, and complexity and selection are measurable through atmospheric assembly. And this is a totally new framework when it comes to life detection on exoplanets. And on the more conceptual side of things, reproduction can be understood as a signature of living systems, whether in the cellular realm or at the planetary scale. And Sarah and I are particularly interested, with Lee actually, in alternate atmospheric configurations that might reflect advanced world that we wouldn't recognize, or that would be much more different from the Earth. So what you see on the top left are atmospheric spectra. This is what we get through our telescopes. And each signature is based on particular molecules that are present in the atmosphere. And so we're very interested in trying to understand what sorts of atmospheres that would potentially be very different from that of Magicos, but still consistent with an advanced and complex world that we might one day see on exoplanets. Thank you very much, and thank you, Claire and the Berggruen Institute, for allowing us to play with these concepts.
Thanks so much, Estelle, and thank you to all of the researchers on this call. I want to just quickly note how radical all of these are together while using the tools of science and technology and AI that we have available today. Sarah and Yarrow on Phaenoterra are revisiting and actually revivifying chemistry that from decades ago that was never fully developed because people for a really long time thought that Venus would be so inhospitable to life it couldn't exist, and so they're really charting a bold path of discovery. Lee, who's a chemist, his work is radical because it's reframing the construction of things that are alive and intelligent and conscious, even from a non-carbon-based perspective. So cool. Penny's planet is radical because it's imagining this very, very extreme environment We've learned so much about what are called extremophiles on Earth now, which has given us like a lot of hope for the kinds of life forms that we might see on other planets. So extremophiles, like in the depths of the Mariana Trench near hydrothermal vents. And so doing this kind of work and stretching the extreme into an even more extreme space is really really exciting. I love that Sam's project and these three co-orbitals. It's almost like Earth is dreaming differently, and Sam is, Sam is kind of the creator of these dreams. And then Magicos, what I really love about Magicos and the work that Sarah and Estelle are doing is that they're not just proposing, okay, like we need way more atmospheric signatures, and then we can say that that's a biosignature. They're completely retooling the category of what a biosignature is to try to look for alien life in a way that we haven't been able to do in the past. Okay, so thank you to all of the researchers. I have more thank yous. So Estelle, who you just heard from, and Ranger Liu, who's now in a PhD program at University of Wisconsin. The two of them were just amazing research assistants. They're the ones who imagined the entire solar system together, and then we world built the planets with the PIs world built the planets. So totally essential in kinds of constructing a system that's gravitationally consistent, and making sure like everything is kind of nothing is exploding. So thank you so much. Speaking of world building, this project is continuing in different media. One that I'm so excited about is a science fiction analogy, and we have some of the writers we've commissioned in the audience. So the scientists have world built a planet geologically, biologically, chemically, technologically. And the writers are going to reimagine that world with characters and how it has a story. So look out for that. This project is not complete. So a lot of what you saw is work in progress, under evolution, as Sam said. I really like that. And we're looking for ways to present the project. We want to do so in scientific venues. And in addition to the sci-fi, we also want to fund more of the science and perhaps even imagine other worlds. Maybe you saw the little piece in the New York Times, I guess, 2 weeks ago on K2-12b. This was first observed by Kepler in 2015, but a closer look with the James Webb Space Telescope indicated a biosignature that they're kind of, it's a little bit mushy. They're hoping to confirm it. And so by imagining like these other kinds of worlds on a planetary scale, how a different star would have influenced all of these planets, how the different like mass, radius, density, chemistry of worlds that are so alien to us than, the 9 planets. I still like to call Pluto a planet, poor Pluto, that we're familiar with. Okay, so thank you so much to Future Humans. Thank you so much to the Institute. And yeah, we will be sending you like a little follow-up, a wrap-up, and this will also be available online. So feel free to get in touch. Proxima-Kósmos.com is where all of the planets live, is actually the right word. Okay. Thank you so much, everybody, and hopefully more soon.